Okay, now let's take a look at purine and pyrimidine degradation. Now first off, let's start out with the easy one. Pyrimidine degradation is very straightforward, and the products of pyrimidine degradation are soluble. So what this essentially means is that we are going to take the pyrimidine ring, we are going to perform some modifications to the substitutions around the ring, the chemical modifications, and then we will be able to excrete this material into the urine. The products of pyrimidine degradation being soluble, that means that they are soluble in the blood and they're soluble in the urine and they can be excreted without a problem. So there are no medical issues with pyrimidine degradation. Now purine degradation, on the other hand, can provide some interesting challenges. The purines, of course, are GMP for guanosine, monophosphate, and AMP for adenosine, monophosphate. Now there are several steps in the degradation of these molecules. The first step for GMP is very straightforward. In this reaction, we are going to release the ribose. So we are essentially cleaving the base off of the ribose sugar of this nucleoside. And the end product is the base alone, guanine. AMP requires a couple of steps. The intermediate steps are not completely important for our purposes, but within these steps we are doing the same process as in degradation of guanosine monophosphate, is we are releasing the ribose sugar. The ribose carbohydrate, of course, can be recycled into PRPP and reused again for nucleotide synthesis. Now the product of AMP degradation, we are going to take this down to the molecule called hypoxanthine. So this is a breakpoint here from GMP to guanine, AMP to hypoxanthine. And now these two processes, these two substrates, converge. And they converge on a single formation of a single product called xanthine. Once we have the product of xanthine, an enzyme called xanthine oxidase will generate the product uric acid. Now uric acid is a general acid, of course. It has a pKa of approximately 5.4. So that makes this essentially a weak acid. Now recall acid-base chemistry is that acids in their deprotonated state, when they have a charge on them, are their most soluble. But as acids become protonated, or associated in their salt form, they become less soluble. And this is the problem with uric acid. Because the pKa is approaching neutrality, we have an ionization state problem with uric acid that we can have too much in the protonated state. Once we have that, it doesn't take much with the concentration of uric acid elevated to cause precipitation. And this precipitation is due to protonation of a portion of the uric acid that's present in the circulation. Now normally, under normal circumstances, uric acid is secreted into the urine. But because of this pKa problem with this acid, if it becomes protonated, the tendency will be to precipitate. This precipitation is increased by temperature drops. And what I mean by temperature drop is it doesn't take very much of a temperature drop. We're talking about a few degrees below our core temperature, such as in our extremities. Hence, precipitation of uric acid forming crystals occurring in our extremities, the major one, of course, being in our big toe, our largest digit on our foot, will lead to uric acid crystals. These crystals, of course, are extremely painful when they come out of solution in the joints. This is called gout, or gouty arthritis. Now there's a very simple method to solve this problem, and that's to inhibit the enzyme xanthine oxidase. The reason for that is because xanthine itself, along with hypoxanthine, are very soluble molecules. Make a note here that xanthine oxidase also converts hypoxanthine into xanthine. So by putting the drug allopurinol into a patient that is suffering from uric acid crystal formation, suffering from gout, we are essentially blocking this enzyme xanthine oxidase and preventing the transformation of hypoxanthine and xanthine into uric acid. By these two molecules being soluble, we can now excrete these into the urine because they are more soluble than uric acid. So allopurinol, which has very little side effects, very well tolerated, is a definite cure for gouty arthritis. Now the other information that we need to discuss about purine and pyrimidine degradation is a process known as the salvage pathway. Now recall in purine and pyrimidine biosynthesis that in order to generate these large molecules, we have a lot of resources going in. So these are biosynthetic molecules that require amino acids to be part of the structure of the ring. Other small molecules, such as carbamoyl phosphate in the cytoplasm. And then we also are going to require a lot of reducing power because we are constructing a large molecule from smaller molecules. And that will take an input of energy. So using these resources, amino acids and other small molecules along with reducing power, we're putting a very, very large amount of energy in constructing the pyrimidine ring as well as the purine ring as well as the pyrimidine ring. Now these ring structures are holding a lot of energy because we put it in there for biosynthesis. But the problem is that when we degrade these, we are degrading them into soluble components that can be excreted. Degradation of these molecules does not mean breaking the ring structure. We are unable to break these ring structures. If we could potentially harvest energy that we put into these structures. But we do not have enzymes that can break the ring. Once we form it, it's there permanently until it's excreted. When we do the degradation process, we are simply removing the ribose carbohydrate, so we can reuse this, and we are modifying the substitutions, the chemical modifications around these ring structures. Yet the ring structures stay. So because there's so much energy put into this, we have what's called a salvage pathway in order to recapture the bases if they are undamaged and reuse them. So for purine salvage, we are going to take the enzyme, the structures, the ring structures, at the level of guanine or hypoxanthine. So recall these structures are from the initial breakdown of our purine nucleotides, where we're cleaving off the ribose structure, resulting in just the base. These bases now, we can use an enzyme that's abbreviated HGPRT, which stands for hypoxanthine, guanine, phosphoribosyl transferase. Now the name of this enzyme is very descriptive. HGPRT is involved in taking PRPP, combining this with guanine or hypoxanthine, allowing us to make the formation of IMP, which is inosine monophosphate. And then recall from inosine monophosphate, we can take several reactions in either direction to make GMP or AMP. And from the salvage pathway, we've been able to capitalize on capturing these bases so we don't have to use the energy to remake them, place them back onto the ribose structure, the 5-phosphoribosyl 1 pyrophosphate, and with a few reactions modifying the ring substitutions make GMP and AMP. Now this enzyme is defective in Lesch-Nyhan syndrome. So this is an inborn error of metabolism. This is a genetic defect, and this enzyme is absent. There is no activity. So because this is not active, we have lost the ability to recover our purines through the salvage pathway. This leads to a very devastating disease of mental retardation and an unusual property of self-mutilation. Another presentation of these individuals is that they develop gouty arthritis. Because the salvage pathway is an important pathway to recover guanine and hypoxanthine, and it also helps prevent the synthesis of uric acid, because we are recapturing these bases on the ribose to make our nucleotides. So individuals that are deficient in HGPRT with Lesch-Nyhan syndrome are no longer able to recapture guanine and hypoxanthine. And so now we are pushing more of these substrates into this pathway of generating uric acid. So these individuals have significantly elevated concentrations of uric acid because of pushing substrates into this pathway, and hence lead to uric acid crystals and gout.